Hello, everybody. Welcome to the week 5 video for the Kombucha Lab. Week 5 will be one of your most busy weeks in lab. So this is a really important week for person A to come prepared, but actually for everyone to read through what you're doing, or else you'll just take a long time and be sad. So for your pre-lab this week, you're going to be learning how to count the microbes and calculate colony forming units per mil. And so there will be some stuff for you to read and watch about that in the lab manual. And then you'll go to the Moodle week 5 and get the week 5 paper pre-lab to print and complete. One thing that's a little bit different about this week 5 pre-lab is that it is entirely on paper. Okay, so normally you have like a Moodle part to the pre-lab and a separate paper part to the pre-lab. This week's is entirely on paper. So it is divided into two parts, part A and part B, so that we can make each part worth 3 points and drop your lowest 6 points. So the two parts are labeled. So here's part A, pipetters, dilutions, and labeling. This is going to be like last week, so you'll already be great at that. Number 2 here asks you to write out directions to make dilutions. This is also something you should have done last week, but it has a couple of twists to it this week. Twist number 1 is all of the volumes have to be less than 1.5 mils, because those little flip cap microcentrifuge tubes are 1.5 mils. And I want you guys to think about dilutions that will fit in those tubes. The second thing is you have to choose a different total volume for each step. And so if you're making 1 times 10 to the minus 4, then you're going to have 4 steps. And so that will require you to have 4 different volumes. Okay, so again, I think I talked about this in last week's video, but just very important to reiterate. I know that making the dilutions can be really confusing, and this is something that you might want to get help on from a friend. It is super extra ultra important that you not write down what your friend has. This will just end in disaster. You'll be sad, they'll be sad, you'll both get zeros, disaster. And so what you'll do is you'll get your friend to explain to you how they did the work. Do not ask them to send you pictures, because this is not helping you to learn. Get them to explain to you how they did it, and then you do it on your paper, but for different sets of numbers, right? So the total volumes that you choose should be numbers that are specific to you, like from your phone number, from your address, from your student ID number, from your sports jersey number, from your whatever. That's all the numbers I can think of. But the numbers that you pick for this number 2 one should be numbers that are specific to you. This will protect you against accidentally turning in copied work and thus getting a 0. So super important that you do not match anyone else in this number 2 section. All right, moving on. Number 3 and 4 here will also be similar to last week. You'll label the plate, and it's pretty self-explanatory. And here we are to part B. And so part B is the stuff that is going to be more new for this week. And so it'll have you to count. It won't have you to count. It tells you the number. So it tells you how many colonies you count, tells you what volume you plated, tells you what dilution you plated, and then asks you for concentration of bacteria in the original sample. In this question, since the original sample is water, this will be colony forming units per milliliter. And so you'll need to change this 100 microliters to milliliters in order to do this problem. There are directions in the lab manual and a video that you can watch for how to do this. Okay. This one is going to be pretty similar, except it asks for a soil sample. And so this is going to be colony forming units per gram of soil, since soil is obviously not in milliliters. Okay, moving on. Here on the last page, it shows you these example pictures, asks you which is the countable plate, and then you count it. Be sure that you get all the little tiny ones. You count the countable plate, and then you do the math to figure out colony forming units per milliliter, or I guess it says soil, so it's going to be per gram. In this case, again, it is going to be super important that you do your own work on this one and don't write down whatever your friend has. Okay, so once you do the pre-lab and you're in lab this week, this is again going to be a very busy week. And so you're going to count your colonies from last week. Okay, that's actually not where you start. I'm not really sure why that's first. Where you start is make observations of your kombucha. So the result person needs to get the jars right away, take off the lids, take good pictures, don't forget the pH strips. The reason that this has to happen first is you do not want the scobies to be bumped before you take the picture. So once the result person gets the pictures, then other people can start to work on the jars. The next thing that you're going to do is freeze soup samples for the TTC assay. So you'll go to page 96, follow the directions on that. But again, you'll do this after you take the pictures. Once you have the TTC samples frozen, then you'll do dilutions and spread plating. So the great news about this is the same thing that you guys did last week. And so last week was your first time to do it. You didn't really know what you were doing. It might have come out kind of rocky. You might have some mold in your plates. But this week is your second time to do it. And so hopefully you'll feel a lot more confident. You'll have a better idea of what you're doing, and it will go both more quickly and better this week. So you'll make dilutions and do the spread plating. At the same time that you're doing this, maybe a different person in your group is going to be making the new kombucha. So you'll remember, you may or may not remember, hypothetically, you remember from week 3 that making both boiling and cooling off the water can take a really long time. And so I would get somebody started on making the kombucha concurrently with somebody else doing dilutions, plating, and labeling the plates. Somewhere in there, when you have somebody who's not busy, then they can look at the plates from last time and practice counting the colonies and calculating. And then whoever is your hypothesis experimental design person will write the hypotheses for this week. So week 5 and week 7, which is basically week 5 again, will be your most busy weeks for lab. So the really cool thing about week 5 is you have only one new thing that you're doing, and it's this count and calculate. Everything else that you're doing in week 5, you will have already done before. And so hopefully this will help you to make it go really, really smoothly.